So it's my pleasure to come here today to talk to you. We just heard a lot about DNA writing, which is super critical because we want to encode information into DNA. It's so powerful, but I'm going to talk to you instead about reading that out. So we've had a lot of sequencing technologies through the years. Obviously we had in the 70s and 80s, we went through Maxim Gilbert, and Sanger came to dominate, especially with Lee Hood's integration into sort of capillary electrophoresis with fluorosequencing of it, and that became very powerful. And then we had Illumina sequencing, sort of SBS-based sequencing, competing with ion torrent, 454, et cetera. But I'm going to talk to you today primarily about sort of third generation or single molecule sequencing. And what's shown here on this slide are two figures from the sort of, I would say, leading platforms for single molecule sequencing currently, which is on the left, you have an Oxford Nanopore Prometheon that's actually in my lab. Note the sort of wood paneled benches that are available at Johns Hopkins University. And then on the right, you see a picture of the Revio floating in a white featureless void, because I don't happen to have one in my lab, but is also a beautiful instrument. Either instrument can now generate on the scale of 100 gigabases of sequencing data for $1,000. I do like to disclose I do have 2 patents that have been licensed to ONT, but the other point I want to feature, which isn't necessarily on this slide, is ONT makes the current cheapest sequencer that's ever existed. It is a $1,000 USB stick that you can plug into a laptop, and you can generate billions of bases of sequencing data from that. For hundreds of dollars, yeah. But you can generate lots of data easily and cheaply. Most of these other sequencing platforms cost hundreds of thousands of dollars. That means that we can now access things. But how does it actually work? So nanopore sequencing has lots of problems. The reason that it was invented or first patented back in the mid-90s, but didn't come to fruition commercially until, let's say, 2014, was that it's hard. The signal that you get is is very rich, because usually most of the pores that we're working with are not 2D materials, but they actually have depth. And so you have multiple bases in the pore at a given time. This means that multiple bases are going to influence the current. And so working with Alex Sementiev, who's still at University of Illinois, I believe, and Jeff Comer, who was a student of his at the time, we tried to simulate what would happen if you had all these different sort of 3 bases, let's say 3, in the pore at a time. And what you can note is that, and this is back about 13 years ago or so, that if you have certain triplets, say GTG, you can easily distinguish them, as shown in green. But if you have a different triplet, let's say TCG, you can't distinguish them so easily. But it turns out that there's an advantage, which is if you can control the translocation rate of the DNA molecule through the pore precisely, you know that after you go from GTC to the next triplet, it's got to be TCA, TCC, TCG, or TCT. So you can actually use a hidden Markov model to eliminate a lot of these sort of other possible methods and actually walk a Markov chain through here and establish Basecalling in this fashion. This was actually how the initial Basecalling worked, and this is a figure from my lab from about 10 years ago, actually from, let's say, I think it's 2014, of part of the Lambda genome. And this is the R6 chemistry from ONT. It's a 100 base pair window as shown in IGV. And you can see there are a couple errors. You have a lot of places where there's a mismatch as shown by a colored letter, deletions as shown by those sort of straight lines. And then there's purple carets in here too that act as insertions. The chemistry, ONT has worked very hard on improving the chemistry, improving the steadiness of the translocation rate is controlled by their motor protein, and getting better detection. But they also have shifted their Basecalling to something called to neural nets. We all know and love ChatGPT. I know I write all my grants with it. Don't listen to that NHGRI. But it's also powerful for doing Basecalling. And so by taking that same kind of data and leveraging that same information, that as the DNA is going through the pore, you get multiple bites at the same apple, you get to see the transit, you can actually get much more accurate Basecalling. So a combination of chemistry and Basecalling has gotten us here, where about a year ago, this is data that we took with the latest 10.4 chemistry, with the latest Basecalling models. And this is the same sort of scale of sequence, 100 bases. It's not the exact same sequence, but it's the same sort of scale of sequence. And you can see that we have many reads which are basically perfect over that scale. Some that have errors, like I'm not making this data up. This is not Photoshop. This is real data. It's just that it's getting pretty good. What else can we see? Well, back, 16 years ago or so, we wanted to see if pushing methylated DNA versus unmethylated DNA through a solid state pore would show any differences. We wanted to understand how methylation, which is a critical feature of the epigenome, could be detected with single molecules. So this is some work we did with the late Kyle Shelton. And what we did was we took DNA that represents something, an important protein DNA binding location, a CTCF binding location, the imprinted control region. And we tried to see what happens when it's methylated versus unmethylated. It turns out when it's methylated, it's actually stiffer, so it can go through the pore at a lower voltage. Others, including somebody sitting right there from University of Washington, and then also a group from University of California, Santa Cruz, Mark Akeson's group, tried to look to see, well, can I actually tell this by just see what's happening by measuring the current as the DNA, as methylated DNA versus unmethylated DNA is going through the pore? And they found, in fact, that you could. Working with Jared Simpson, we then followed this up with the sort of more mature ONT platform, and we demonstrated using a hidden Markov model, again, that we could actually call methylation using the shift in current signals, as you can see here, from GCT TGA to CGA to methyl CGA. You can distinguish all three of those possible k-mers. It was a 6-mer at the time in the R9 pore. I will note that my good friends at PacBio can also detect this based on how much the polymerase pauses when it hits this methylated mark, because methylation does stuff. You know, it's changing the local hydrogen bonding structure. It's changing how the DNA fits in the enzyme. It's changing how the enzyme reacts to it. It's changing how current can flow through the pore around it. So we can actually measure methylation with both of these single molecule platforms. I know I featured more than nanopore stuff, but either platform can actually detect it. Well, what does that mean? Why do I care about measuring methylation? Well, first of all, it turns out that the allele you got from your dad and the allele you got from your mom sometimes have different methylations. Shown here is a region of MEG3, which is a long non-coding RNA, and that's known to be monoallelically expressed. And part of how that's regulated is that methylation is different on the two haplotypes. Now, you might say, well, I could read that with short-read sequencing, couldn't I? Well, you could, but you'd have to run into a heterozygous SNP that would distinguish your mom's copy from your dad's copy. Otherwise, it would just align to the human genome, and you wouldn't know which copy you're looking at. With long-read sequencing shown with PacBio on the top and with ONT sequencing on the bottom, we see the same behavior where the red indicates methylated and blue indicates unmethylated. You can clearly see that haplotype 2 is unmethylated at both these locations. And again, this is a CTCF binding site. It's part of a regulatory region that's actually helping to control what's happening. Even if we don't run into a SNP, though, I'm going to argue to you that the methylation is useful, and I can cluster based on methylation alone. So I'm cheating here. I took some HCT116, which is a colorectal cancer cell line, and HC002, which is the genome in a bottle lymphoblast cell line. And I mixed the sequencing data from them together, and I looked at this region around RIN3, the CPG island, as you can sort of see here, and I clustered it based purely on methylation of a sort of 20 kb long region. When I did that, I got this result, where based only on the methylation, I found 4 different clusters. 2 clusters which correspond to HCT116, 2 clusters which correspond to HC002. And so I can actually see the alleles and what cell type they are falling out of the data naturally just by looking at the methylation. I'm not using SNPs, I'm not using genomics here. I'm only using methylation to be able to distinguish the haplotypes and the cell types from each other. So cell type deconvolution, that's an interesting idea, and that's something that might be powerful, we thought. We're not the first people to think so. Even back in the microarray data era, Hausman et al. And others have used methylation signals to be able to figure out from, say, whole blood, what the distribution of cell types are. Because different cells in your body have different epigenetic signatures. And folks like Joe Ecker and Tommy Kaplan have generated these big methylation atlases, which are really useful to people like me who are very lazy, to be able to just look up and see, oh, what does that cell type's methylation profile look like? And then be able to use that to look at my reads. So my student Luke Marina wanted to see, well, how well can we do this with nanopore sequencing alone? How well can we actually count how many different cell types we have with nanopore sequencing alone? So he took some PBMC samples And he did targeted sort of nanopore sequencing using a method called adaptive sampling, which I'm not going to go into at this time. And he also did single-cell RNA sequencing, sort of as the gold standard, because he figured that single-cell RNA sequencing would tell him exactly what the different cell type proportions are in his sample. Did this on 3 different samples, generating something like 100x coverage at the regions of interest that he had for DNA sequencing, and sequencing something on the order of tens of thousands of cells from the single-cell RNA sequencing. So that was kind of fun. And what he found was that he can, in fact, distinguish, and he gets pretty good results where he's seeing like the proportions of B cells, monocytes, macrophages, natural killer cells, T cells that are present in his sample. He can actually distinguish them, and they sort of agree between methylation and single-cell RNA, not perfectly, but you might say, well, the single-cell RNA had tens of thousands of cells, and the single-cell and the nanopore sequencing only had about 100x coverage. So it could be a bit of a sampling error there. It could also be that methylation is telling us something different than what the transcriptome is telling us. We're not necessarily quite sure what to make of this yet, but we think it's really promising to be able to phase out and separate out and quantify from a cheap sequencer that's $1,000 instead of $100,000. I can't believe there's still a $100,000 flow cytometer. You could imagine having $1,000 a MinION and do adaptive sampling and be able to figure out what the proportions of cells you are in some tissue sample. But there's more than that, because we can actually cluster the reads. So shown here is sort of uncluster those PBMCs, and then on the bottom, we're actually clustering out the reads that belong to the T cells. What does this mean? Well, that means we might be able to actually phase to see, well, I have this cell type specific DMR. How far is it to a gene? How far is it to a mutation? How far is it to something else that I want to connect and see what the state of that is in some cell type from a bulk sample? Because all too often, we're not taking our samples that we're getting from human tissues or mouse tissues or animals and flow sorting them carefully into single cell bins. Instead, we're just sequencing it in bulk. And so what I'm arguing is that we can later deconvolve out and see which reads belong to which cell type, and then see, maybe I got somatic mutations that arose only in this cell type or only in that cell type. And it turns out that with the read lengths that we can achieve pretty easily with single molecule sequencing nowadays, say 20 kb long reads, we are often going to run into a gene or a promoter or an enhancer or something that we want is usually within range of a cell type specific DMR. So we might be able to use this to look for mutations in genes. We might be able to use it to coordinate and see, if I know that this is in this cell type, what's going on with this enhancer in that cell type? Is there a change in that cell type in a cancer sample or in some other tissue samples? So we think that that might be interesting too. And then as a sort of final vignette, we weren't satisfied with just looking at methylation with 5-methylcytosine. We wanted to also look at hydroxymethylcytosine. So we've heard about, you guys, some of you here have heard me talk about hydroxymethylcytosine a couple times in the past. And I know that some people like to try and look at it in cell lines, but I'm here to tell you, I don't think that that's the right way to go. I think that there is not a lot of hydroxymethylation in cell lines because cell lines are actively dividing. Hydroxymethylation is an intermediate along a demethylation pathway, which I do not think is at high levels in anything that's mitotically active. So what I instead chose to do was get from my best friends in the whole world at JAX mouse tissue, because it turns out that you can just order tissue directly from them. You don't even need to have like a mouse protocol. You can just get the tissue. They'll take care of harvesting the tissue. I spent enough time doing mouse work during my postdoc. I didn't want to go back there. So we got some mouse tissue from JAX of cortex, cerebellum, heart, and liver, of F1, so we'd be able to phase of black 6 and SJL mice. And then we did both long read sequencing, and we're doing short read EM-seq from NEB, and their sort of modulation that lets you look at hydroxymethylation with EM-seq. The first thing I'll tell you is that the nanopore model, I know my friend Dongfang has looked at this and has found that it does make mistakes with HMC. He's right. But broadly, regionally, we do see pretty good correlation with EM-seq, as you can see here in this cerebellum-specific gene. On the left, you can see ADAM22, and the liver over on the right, you can see enrichment of HMC in CYP3A type genes in the liver, but not in the brain samples. We see the same sort of global levels of methylation reported from both short and long, and we see actually significant levels in here. If you look at 002, HC002, you will not see very much HMC. But if you look at mouse tissue, this could be a sort of benchmark or tissue in a bottle, so to speak, that we can use to validate some of these methods to look to see what's going on with HMC. And here's why I'm going to tell you why this is important. If you look using, say, bisulfite sequencing or just looking for modifications to the DNA molecule, this is what Atom 22 looks like, the gene. And when you're looking at, this is sort of condensing our 5 replicates into like one line just for ease of visualization. And you can see cerebellum, cortex, heart, and liver. This is what this looks like. You see basically no difference. If now I split it out according to whether it's HMC or MC, so the MC is now the solid lines, the dotted lines are the HMC, you can see very clearly that there's a difference between the brain samples and the heart and liver. That was not visible with traditional sort of just looking at modifications. So it could be important to be able to look at this because it's just the cell, I'm anthropomorphizing here, but the cell wants to change something about that gene. This gene is important, and where it's positioned is important for sort of membrane potentials and things like that. So it could be important for brain development. Here's another great gene, ApoB. We all know ApoB is really important in metabolism and things like that, and it turns out ApoB has enrichment in the liver of HMC. It could be that this could tell us more about how epigenetic regulation is actually being modulated in tissues and in reality that we cannot observe because it's just the cells are dividing too fast and the HMC marks get wiped away because they're too transient. But in cells in actual tissue, the mitotic clock speed is slower often. We also think that you're going to be able to say the same kinds of things by looking at non-CG methylation for similar reasons. We want to look at tissue because it will give us a more realistic appreciation of how epigenetic regulation might be happening, even though it's also very complex because of the heterogeneity of tissue. With that, I'm happy to take any questions. I want to acknowledge other people who have worked on this, especially Luke Marina and Paul Hook. And my funding sources, of course, my friends at NHGRI, the rest of my lab, and thanks.
Questions for Winston? Can you say a little bit more about the 5-HMC? It's not something that I think folks are
Sure.
As familiar with.
Yeah, so I wish I had that, my little chemistry cartoon. It's on my poster tomorrow. So the HMC, so normally you have cytosine or 5-methylcytosine. What happens is there's this TET enzyme where to remove methylcytosine, TET enzyme will then oxidize to make it, instead of methyl, it'll make it hydroxymethyl. So a CH2 and then an OH, right? This is no longer recognized by DNMT1, which is the sort of methyltransferase that maintains methylation marks, but also it can then be further oxidized to carboxyl or formyl methyl. But the point is that it's part of this demethylation pathway. It's part of the way that the cell is trying to remove methylation from this gene, from this location, if that makes sense. Thanks, Mark, for the clarifying question.
Thanks. Okay.
It's a great talk, Winston. I had a question just about the cell-to-cell variation that you see in methylation and how that relates to the methylation detection error rate. Like, are these variations real, or are they artifacts of the data? And how important?
So are you saying
are they biologically
within a read, if it says it's methylated versus not methylated, or are you saying if I can detect this?
Yeah, I mean, some loci are pretty much always methylated, but some have some variation. And is that just that location that's hard to see if it's methylated or not, or that location?
Yeah,
it's biologically variable.
So to restate your question, how well can we call methylation, and is there biological variability in methylation?
Yeah.
Yeah. So we are filtering to only accept really confident calls here in methylation. So if we're not sure about it, we would drop it. I will tell you that from looking at bisulfite data and other data and looking at methylation data for, 15, 20 years now, that there is biological truth to the fact that you do have some regions of the human genome or mouse genome or mammalian genome where you have a lot of variability in methylation. This is also termed by some methylation entropy. There have been a bunch of folks, like Amos Tanay has looked at this with epi-alleles, as has Chris Mason, and tried to say, what can we see from this? It turns out that you can even track clonality. So if you look at your colon and you look at different crypts in your colon, you can figure out which cell belongs to which colonic crypt because of the noise in the methylation of the stem cells then gets relatively replicated up through the whole crypt. So some of that is real. Is it biologically meaningful or functional? That's a different question. When we're trying to cluster according to cell type here, we're generally looking regionally. You know, we can smooth out small variations because we're looking regionally to see, oh, this whole thing is different in this area. But I would also direct your attention, there's a tool called PoorMeth, on the variability question, there's a tool called PoorMeth2 where they try and establish, can you see differentially methylated regions, but differentially methylation entropy regions by Alberti Maggia in Italy. And I think that's also a powerful approach because noise in methylation might tell you something about that the cell is not regulating its genes so well anymore. So in cancer, for example. It was a long-winded answer. I hope that was helpful.
Good answer. Thanks.